topic of this video is an introduction to enzymes. We're going to be talking about what enzymes are, how they work, and how they're different from ordinary chemical catalysts. So to recap, in the activity for Wednesday, you were introduced to catalysts. And catalysts are substances that increase reaction rates without being consumed themselves in the reaction. And the way that these catalysts work is that they speed up reactions by lowering the activation energy. So let's look at this diagram here. So we have one reaction shown in red that's uncatalyzed. So that has some activation energy associated with getting from the reactants to the transition state up here. All right. But then in the blue line, now we've added a catalyst. And we can see that in the presence of that catalyst, we have reduced the activation energy. It takes less energy when the catalyst is present to get from the reactants to the transition state. And because there's a lower activation energy, that ends up speeding up the reaction rate. And that can be seen through our Arrhenius equation. Right? Lower activation energy, faster rate. Right. And we said on Wednesday that there are two different types of catalysts. Both of these speed up reactions. Both of them do this without being consumed themselves in the reaction. So those types are heterogeneous. These are present in a different phase than the reactants. And homogeneous catalysts, which are present in the same phase as the reactants. So that's where that homo prefix comes in. So hetero, different, homo, same. All right? So that's kind of a general summary of catalysts. But now, since we're in a foundational biochemistry class, the next question to ask is, okay, so how are reactions catalyzed in biological systems? And it turns out that nearly all biochemical reactions are catalyzed. And they're catalyzed by these molecules called enzymes, which are homogeneous catalysts. So they're present in the same phase as the reactant. Present in the same phase as reactant. And like all catalysts, the way that enzymes function is they increase the reaction rate by lowering the activation energy of that reaction. So what are enzymes? Enzymes are usually proteins, but they can also be RNA. So some RNAs can form what we call ribozymes, which are basically just RNA enzymes. And a really good example of this is that one of the major reactions involved in protein synthesis is actually catalyzed by an RNA in the ribosome, not a protein. Right? So that's kind of an overview of what biological catalysts are. But there are some really important differences between biological catalysts, enzymes, and normal chemical catalysts. The first one is that enzymes result in faster reaction rates than an ordinary catalyst. Enzymes can actually increase the rate of reaction up to 10 to the 12 times what the uncatalyzed reaction rate is. So that's a huge acceleration in the reaction rate. Ordinary catalysts can't achieve that kind of an acceleration. All right, so that's the first way that enzymes are different. They're also different because they can achieve these huge accelerations in reaction rates under pretty mild conditions. Life has to happen under very specific conditions. Low temperature, so Human body temperature is 37 degrees Celsius. Human body temp. Which is quite a bit below the boiling point of water. So we're at a pretty low temperature. We're at near neutral pH. We don't need to add any strong acids or strong bases for this to happen. And we are at 1 atmosphere of pressure. So we're not getting really compressed by high pressure when these reactions are happening. So they're able to achieve these huge changes in reaction rate under pretty normal conditions. And in addition to that, they're highly specific for both the reactants, which we're going to be calling substrates. Substrates refers to the reactants of an enzyme catalyzed process. So they're highly specific for their substrates. They're also highly specific for their products. All right. That's pretty different from ordinary catalysts. Ordinary catalysts can often have side products where something else is made in some amount in the process of trying to make the thing that we actually want to make. Enzymes don't have that problem. They are almost always making exactly what they're supposed to make. And that's that specificity piece. The other really unique thing about enzymes is that they can be regulated by other substances that aren't involved in the reaction. Okay, so that's a bit about why enzymes are so special, why they're such unique catalysts. So let's dive a little deeper. So first, we need to do a little bit of nomenclature. So the way to know that you're looking at an enzyme is to look for this ASE ending. If you see that ASE ending, you know you're looking at an enzyme. And enzyme names, for the most part, are going to tell you what they do. You'll see the substrate, and you'll see some kind of description of the reaction in addition to that ASE. All right, so here I'm showing you this example of lactate dehydrogenase. So let's break this down. So we see the word lactate, okay? That's going to be the substrate. Because that doesn't really have any, like, reaction type of word in it. It's just lactate is just a molecule. So that's the substrate. And then we have this dehydrogenase. All right. So we see this ASE, we have an enzyme. Now, let's look at this dehydrogen. So this enzyme does exactly what that dehydrogen would imply that it does. It's going to remove hydrogens from lactate. All right. Literally dehydrogening lactate. So you can look at this, at these structures here, see that lactate has these 2 hydrogen atoms, and then pyruvate, what it's being converted to, has none. So we are losing hydrogens. The enzyme is removing hydrogens. It's dehydrogening lactate, right? So that's kind of the naming, and there are 6 major classifications of enzymes that could appear. And those are classified based on the types of reactions that they perform. So like, what are the actual reactions that are being performed? All right, so that's nomenclature. Let's talk a little bit about that specificity piece, because there's a lot to that. So we already said enzymes are highly specific for a particular reaction, or in some cases, a small subset of reactions. And the way that they achieve this is that they have very highly specific active sites. And the active site is where catalysis occurs. So these are typically some sort of pocket on the enzyme surface. And what makes these so unique is that the chemical groups in the active site are arranged to be complementary to the groups on the substrate in shape. So that's this geometric piece. And also in charge and polarity. So that's this electronic piece. And so substrates with a complementary shape and chemistry can bind to the active site. So substrates can bind if the shape and chemical groups are complementary to the active site. So here you can see we have this active site with this very specific shape, and we have these little chemical characteristics that they're pointing out here. And then the substrate has a shape that's very complementary to this shape. Like that looks like it's going to insert right in there and be able to fit kind of perfectly. All right. So that allows the substrate to enter the active site. Once they're in the active site, they will bind based on the chemical groups that they have via intermolecular forces. So that's the same intermolecular forces that we were talking about last semester. Hydrogen bonds, ionic interactions, London forces, and dipole-dipole interactions. Right? And if the shape and chemistry don't match up, they won't be that molecule won't be able to fit into the active site, and it won't be able to make those intermolecular forces to hold it there, so it won't bind. And so you might see this called the lock and key mechanism of enzyme specificity. So basically you can think about the active site as a lock and the substrate as a key that fits into that lock. All right? Something else that came up in chapter 11 was that enzymes are able to differentiate between mirror image molecules, all right? And this is a concept called chirality, all right? And this occurs when a carbon is bound to 4 different groups. So chirality occurs when a carbon atom is bound to 4 different, that's very important, groups. So let's look at this molecule here, all right? So as it turns out, there are 2 non-equivalent ways to draw this molecule. And where that comes from is this carbon right here, which is bound to this group, and this group, all right? And it's also, we have an implied hydrogen. So it's bound to 4 different types of groups. So let's think about how we can draw this. So we can draw this like this. So important note is that we're going to keep this group, which is this group, we're going to keep that on top as a point of reference. That's going to come down to the carbon. I'm going to keep this group in the plane for now. So right? And then we're going to put our NH2 coming out of the page and our hydrogen going into the page. All right, so that's one way to draw this. We can also draw this like this. So everything's going to stay the same, except we're going to put the hydrogen coming out of the page and our NH2 going into the page. All right? And so these are not the same molecule. They do not overlay. And let me prove it to you. So let's test it out with this one. Let's rotate it. All right, always keeping this in the same place. So we're going to rotate it once. So then this group's going to become the hydrogen. The one going into the page is going to be this group. And then the one coming out of the page is going to be our NH2. All right? So you can already see that this is not the same as this one, even though we have our NH2 coming out of the page. Let's do it let's do it 2 more times. So if we do it 1 more time, then we end up with our NH2 in the plane, our hydrogen pointing back into the page, and this group coming out at us, right? So that's still not the same as that. And then 1 more time. We end up back where we started, and we never had a version of this that we rotated that looked like this. So these don't overlay. These are called chiral, all right? And most of the time, a reaction is going to make some amount of both of these. Enzymes are unique. Enzymes are able to differentiate between these because the way that their chemical groups are arranged means that only one version of this molecule would have the correct orientations to be able to have all the interactions that are needed for that molecule to bind to the active site. All right, so you can see that here. So here our groups are very specific, and they are looking for very specific complements on the substrate that's binding. All right, so if any of these is not in the right place, we're not going to be able to get that interaction forming. That molecule is not going to bind. All right, and so these mirror image molecules are called stereoisomers. And because of the way that enzymes are set up, they can differentiate between stereoisomers and select for a specific one, allowing them to make very specific products and make sure that they're making exactly what they want to make. So that's enzyme specificity. The last thing to know is that some enzymes require cofactors to function. And a cofactor is anything that binds to an enzyme and helps catalyze the reaction, but is regenerated. All right? So bind to enzyme and help catalyze the reaction. So these can be metal ions like magnesium, zinc, iron, or copper. This is why we need some amount of all of these in our diets. It's so that our enzymes can function. It's also why metals like cadmium and mercury are toxic because these are in the same group on the periodic table as zinc, so they can actually bind to the same proteins as zinc, but they can't carry out the same reactions, so they end up inactivating the enzyme. So that leads to these two types of ions being toxic to our cells, right? So these metal ions can be very important, that you're getting the right one bound to the right protein. In addition to metal ions, we can also have coenzymes. These are carbon-containing molecules. And there are two types of coenzymes. Co-substrates, which, as the name suggests, they bind to the enzyme with the substrate. So that would be something like NAD plus in this reaction. All right, so ethanol binds, NAD plus also has to bind, and only when we have both of those will the reaction occur. All right, so that's co-substrates. Prosthetic groups are coenzymes that are permanently associated with the protein. So this is something like a heme in hemoglobin. All right, that's always in hemoglobin, but it's not, and it's participating in the reaction, helping to carry the oxygen, but it's not actually being changed in an irreversible way by the reaction. That will get regenerated later, and that is the key feature of coenzymes, is that they are able to be regenerated so that they can be reused to help catalyze another reaction. And that is an introduction into enzymes. In the next video, we're going to talk about enzyme kinetics. We're going to talk about what they are, why they're useful, and how we can determine them.